Hi students, welcome to Science Extension and Module 1, the Foundations of Scientific Thinking. Once again, we're going to try and cover a couple of different concepts within the one video, which I will split up into smaller sections, because this time we want to have a look at some of the ethical frameworks that may impact on scientific research. So we want to analyse the influence of ethical frameworks on scientific research over time, including but not limited to human experimentation, experimentation on animals, biobanks, and the use of research data. The second area we want to have a look at in ethics is around animal experimentation. Animal models in research have historically played a pivotal role in advancing the scientific understanding of the living world. Humans have benefited from animal-based research, and generally, research involving human experimentation is conducted first with animals and then, if it is deemed to be safe, with human volunteers. For example, animals are used in research on human diseases, studies related to addiction and drug abuse, and on the effects of space travel. Despite many years of animal-based experimentation, the use of animals in research continues to be a topic of public and scientific debate. The scientific rationale for using animals in research. Worldwide, several national committees have examined the use of animals in research. There are substantial grounds for justifying animal research, particularly those that are focused on benefiting humans. For example, the Nuffield Council on Bioethics concluded that because of evolutionary continuities in the form of behavioural, anatomical, physiological, neurological, biochemical and pharmacological similarities between animals and humans, there are sufficient grounds for the scientific hypothesis that in special cases, animals can be used for models to study particular aspects of biological processes in humans and to examine the effects of therapeutic and other interventions. Three main types of research that involve the use of animals include research that advances scientific knowledge, for example, understanding genetic, physiological, or biochemical mechanisms; research that uses animals as models to study the mechanism of human disease and to develop interventions; research that uses animals in toxicity testing. The biological similarities between humans and animals used in research imply that those animals may possess certain traits, for example, sentience, higher cognitive capacities, the capacity to flourish, sociability, and the possession of life, that make their use morally troubling. The mistreatment of animals in the name of scientific research is another area of concern. In many countries, the use of animals in research is regulated by laws and statutes. From the 18th century onwards, several countries enacted laws to protect animals, such as the 1876 Cruelty to Animals Act in England. In Australia, animal welfare legislation is enacted and regulated by its states and territories. In New South Wales, the ethics guidelines concerning the use of animals in research are derived from the Animal Research Act 1985 and the Animal Research Regulations 2010. In reality, many people now choose products actively and favouring those that are not using animals in testing. So what's the role of animal ethics committees? Animal ethics committees at research institutions are responsible for administering regulations relating to the ethics and morality of using animals in research, including the necessity of such research and the right to use animals for the benefit of humanity. Animal ethics committees review every research proposal that uses vertebrates, whether they be for research or educational purposes. Some of the questions that guide their review are: what are the goals of the research? What's the probability of success? What animals are to be used? What effect will there be on the animals used in the experiment? Are there any alternatives to using animals in the research? In Australia, the National Health and Medical Research Council has produced guidelines that researchers must adhere to. Institutional animal ethics committees evaluate research proposals using these guidelines. The following list summarises the main guidelines for animal research in Australia. Firstly, respect for animals must underpin all decisions and actions involving the care and use of animals for scientific purposes. This respect is demonstrated by using animals only when it is justified, supporting the well-being of the animals involved, avoiding or minimising harm, including pain and distress to those animals, what is likely to happen to the animals during the project, including adverse effects from husbandry, supply, transport, and procedures, applying high standards of scientific integrity, applying replacement, reduction, and refinement, known as the 3 Rs, at all stages of animal care and use, The replacement of animals with other methods, for example, cell cultures, tissue banks, abattoir materials, the reduction in the number of animals used, and the refinement of techniques used to minimize the adverse impact on animals. And secondly, knowing and accepting one's responsibilities. Adherence to these principles are monitored and implemented by the relevant institutional ethics committees. One interesting example of animal experimentation is the zebrafish model of Duchenne's muscular dystrophy. One particular research project that involved the use of animals was carried out by Dr. Don Love and his research team at Auckland University. They were using zebrafish as a tool to study heritable human diseases such as Duchenne's muscular dystrophy. The mutated zebrafish are genetically modified to express the muscular dystrophy condition. Zebrafish are native to the slow, warm, fresh waters of the Ganges River. They're easy to maintain and propagate in the laboratory. They're inexpensive, and much of the embryonic development is visible with light microscopy. Zebrafish are an example of a model organism. Model organisms are non-human species used by scientists to study the biological processes behind many systems in humans. Model organisms exhibit genetic, molecular, biochemical, or physiological similarities to humans. By using model organisms such as zebrafish, scientists can understand many essential aspects of human conditions before working on humans, such as developing treatments. Duchenne's muscular dystrophy is a neuromuscular disease. It results in progressive muscle degeneration and weakness. Initially, it affects the voluntary muscles, causing weakness and dysfunction. Later, involuntary muscles degenerate, for example, the diaphragm and the digestive system, resulting in the death of the affected individuals. Currently, there's no cure for Duchenne's muscular dystrophy. Muscular dystrophy is caused by a defect in the gene coding for dystrophin proteins expressed in muscle cells. In humans, the dystrophin gene is found on the X chromosome and is recessive. This means that the disease is sex-linked and primarily affects boys. Females with one copy of the mutated dystrophin gene are carriers and do not express the condition. Females with two copies of the mutated gene will develop muscular dystrophy, and it affects approximately 1 in 3,000 live male births. Since it's not possible to experiment on humans, scientists use the zebrafish model of the disease to understand the condition better. Normal zebrafish possess the dystrophin gene, which is very similar to the dystrophin gene in humans. To create zebrafish models, scientists use a genetic technology called RNA interference. As a result of this genetic manipulation, those zebrafish cannot express the dystrophin gene and thus develop muscular dystrophy. Fish with the disease are easily identified because they cannot swim normally. Since the zebrafish is a vertebrate animal, the research project must be approved by an animal ethics committee before it can be carried out. Before approving the research, the ethics committee must be convinced that the value of the outcome of the research must be greater than the suffering imposed on the animals. Furthermore, the researchers must demonstrate that they will take all measures to minimize any unavoidable suffering. This is estimated against a standardized scale of degrees of suffering. During the research, the well-being of the fish is carefully monitored and dealt with humanely. In this case, the Duchenne muscular dystrophy research using zebrafish, the Animal Ethics Committee at the University of Auckland approved the study because of the ultimate potential of the work to lead to treatments for Duchenne's muscular dystrophy.